outra coisa que eu queria dizer para vocês é que, na realidade, essa conferência vai ser dada por mim e pelo Dr. Boro Doprulite, que está aqui, presente. O Boro é o CEO da Caring Cross, que vocês vão ver ao longo da apresentação. Ele vai apresentar a Caring Cross, já que todos os slides que eu tenho são dele e da Caring Cross. E ele se formou na Austrália em microbiologia e patologia, depois fez um PhD em virologia, depois ele foi para o NIH, onde ele foi Fogarty Fellow, fez 2 pós-docs no NIH, depois ele foi para John Hopkins, não pode encostar na mesa, e depois da John Hopkins, as he says, he joined the dark side of the force. He then went to private companies. He created a number of private companies, but he's actually not the dark. He is the good side of the force, because Caring Cross is actually a non-profit organization, and as you will see during his presentation, its mission is to really lower the price of those advanced therapy programs in low and middle income countries, and also in the high income countries of the world. And now I can speak in Portuguese, because the other person I want to introduce is Professor Marcos de Lima. Marcos de Lima fez a faculdade de medicina dele aqui na UFRJ, se formou em 1986, onde ele fez a residência também e o mestrado. E ele aí foi para os Estados Unidos. Infelizmente, perdemos ele para os Estados Unidos, como já perdemos várias outras pessoas. Mas, felizmente, ele mantém o vínculo aqui com a nossa terra. Ele foi, nos Estados Unidos, ele foi professor no MD Anderson primeiro, depois na Universidade do Texas, depois na Case Western, talvez não nessa ordem, mas atualmente ele está na Ohio State University e ele é um dos maiores especialistas na aplicação de CAR-T, de células CAR-T em pacientes, né? E ele é uma peça importante também dessa colaboração que a gente desenvolveu nesse programa com a Fiocruz, e vai nos ajudar muito nos ensaios clínicos de CAR-T aqui, e vocês vão ver essa parte também ao longo. Mas ele diz que o principal título que ele tem, na realidade, é ser cidadão e natural de São João del Rei. Ok? Bom, since Boro doesn't understand, perdão, como o Boro não entende português, eu vou falar em inglês. Meus slides estão em português, os dele, obviamente, em inglês, e aí eu chamo ele para falar sobre a Caring Cross. Tá? So, this is the title, the Advanced Therapy Programs in Fiocruz, and I just want to let the younger people know that this project started in June of 2022, okay? I retired here, I think it was signed a month before my retirement, and then at that time, Nízia was still the president of Fiocruz, and I had a meeting with Nízia, Mário Moreira, the current president, and Marco Krieger, and they invited me to coordinate an advanced therapy program at Fiocruz. So, the idea is written in Portuguese there, you can read, is really to implement advanced therapy program and make this fit into the budget of the Ministry of Health. And not only the Ministry of Health, but also the insurance, the private health insurance. And so the presidents Fiocruz at that time decided to create this program of advanced therapies. The name is awful, Fio PTA, but I think they need a marketing person there. And the main objective is to create in the country the scientific and technological basis that are needed to furnish or advanced therapy products in our public health system. So again, to accommodate with the internal politics at Fiocruz, we actually increased a bit the reach of what we called advanced therapy program, advanced therapy products. And we listed as products of gene therapy, products of cell therapy, products of tissue engineering. And that would strictly be what is currently called advanced therapy medicinal products. But we included products for immunotherapy and therapeutic proteins in this program created at Fiocruz. We had to write termo de referência, and then Camille Sacchetti, who is also here, she is related to the Ministry of Health. She directed DECIT for during the dark years, and we owe her a lot because thanks to her, we had the vaccine at Fiocruz. And we together wrote this TR. You all know termo de referência for this program. So this is a joint work with Camille. And why is that? So it's basically, those are really revolutionary therapies. They can offer cure to uncured diseases. And in theory, it's a one-shot therapy. And the big problem is, people developed, like Boro, developed the science and technology. This was then, a lot of the science and technology developed was appropriated by the universities, and then as the American model goes, people create startups. Those startups are bought by big pharma for billions of dollars, and then they have to recover the investment. And the prices charged are really, I would say, shameful. And to give you an idea, just in the States, they think that the financial impact of those gene therapies is going to reach 25 billion a year. Okay? And at the time we compared, and Camille got the data, this would be the whole SUS budget. Okay? And the idea was then that Fiocruz had to play a role in decrease the impact of those PTAs. So to give you an idea of the financial impact, those are the 7. I just got the updated slide from Camille, because the one I had went through 6, but now recently in 2024, we had the 7th one approved by Anvisa. And you can see the names, and you can see the prices set by CEMED. And for those of you who do not know what CEMED is an official organ, and then Camille can correct me because I'm not very familiar with the whole thing, but that sets the price for those PTAs in Brazil. So the price set by CEMED for Luxturna is 1.9 million. For Zolgensma, por trás. For Zolgensma, for muscular spinal atrophy, is 7.6 million in reais now. And it's already incorporated in the public health system. And then you can read the Kymriah, Carvykti, Yescarta are all CAR-T cell-based therapies. The price is in reais. Tecartus also is CAR-T. It has no price still by CEMED, but the price in dollars is 370,000. And just now they approved the Roctavian for hemophilia A, and the price is $2.9 million, okay? And more are coming. And the other ones that are coming are listed here that are already in process of being registered in Anvisa. It's Elevidys for Duchenne at an anticipated price of 15 million reais. And that's based on the international price, okay? And again, the source is Camille. And Upstaza for this deficiency in this enzyme, which should be priced at $18.6 million. So we clearly need other options. And just to give you an idea, I took this from the Anvisa homepage. You can see the BioMarin hemophilia A product was with a clinical trial already initiated. And there are about 23 clinical trials being performed on basically gene therapy, maybe 2 or 3 in cell therapy. So it's what we're calling at Fiocruz a tsunami that's going to hit the market. So the objectives of this program encompass a lot of things. There are many axes that we devised, and again, I want to acknowledge Camille's contribution to this. So the first one is infrastructure, and the Ministry of Health had already supported a number of cell technology centers throughout Brazil, and the idea is that, you know, this investment is not wasted. But there is a need for projects to structure the production of those PTAs under good manufacturing practices, ou BPF, boas práticas de fabricação. Then there is another axis, which is research and development. The idea at that time was that Fiocruz would lead a process that would include not only Fiocruz, but other places, because Fiocruz recognizes that it doesn't have a very strong tradition in chronic degenerative diseases. As all of you know, Fiocruz is tradition is in infective diseases, and we owe a lot to Fiocruz during the pandemic. The idea would then to support projects in gene therapy, cell therapy, in the Brazilian genome, which was, I should say, inaugurated in Ribeirão Preto on Monday with the presence of the Minister Isia Trindade, and also the development of new biomarkers and diagnostic kits, not only for rare diseases, but also for neglected diseases. Within this axis, Fiocruz just launched a program which is called Innova in Advanced Therapies with a budget of 20 million reais, and this was only internal. We're fighting that the next round, Fiocruz launches a program in advanced therapies that's both intra and extramural. And I think the whole success of this program depends on having extramural call for applications. And in this one, I think there were about 42 applications, if I'm not wrong, internal. Then there is an arm about innovation and production. I'm not going to read that, but you can see there that the CAR-Ts are part of it in short time. There is also, you know, the RNA vaccine platform that is well advanced at Fiocruz, probably going into clinical trials very shortly. Then, you know, human resources, capacitação. Then the part of, you know, intramural and extramural that I do hope I convinced the higher hierarchy at Fiocruz that without the extramural support, this program won't work. And then the regulatory, which I'm going to talk to you a little bit later. And the regulatory part of it is also fundamental for the program. So why was this really a clever decision made by the presidency and especially the vice presidency in production and innovation in health? Because there was a huge investment at Fiocruz for the development of the vaccine for COVID-19. And that's a viral vector-based vaccine. So a lot of the infrastructure was already built. And so using this infrastructure to bring now viral vector production at Fiocruz for both CAR-T cells, and you're going to see for neurodegenerative diseases later on. I mean, for ex vivo and in vivo gene therapies made a lot of sense, okay? So now I turn to Boro to talk about Carrying Cross.
Thank you, Antonio, and it's a real pleasure to be here. And I just want to state categorically that if it wasn't for Antonio and Camille, we wouldn't have this kind of a program. So really, I just want to make sure that they're acknowledged as the initiators of this program. And we're very excited to initiate the program where the goal here is to technology transfer the knowledge that we have. We've been in the field for about 30 years. It's not just myself, it's my entire team. And we've started companies together. We've gone to different places. And then when I came up with the idea of a non-profit as a way of enabling affordable and accessible therapies, they wanted to join me again. So the old team is kind of back together again. We had an impact on the very first lentiviral vector clinical trial. We also, the vector that resulted in the first FDA-approved product, Kymriah, that was the vector that we developed for the University of Pennsylvania. So we have a long history. And as Antonio stated, that when we saw that the therapies became commercial and the prices that they were charging for it, we thought it was kind of ridiculous. There has to be a better way of delivering these therapies. And so we thought that working more directly with hospitals, with public health systems, you don't have to have a pharmaceutical intermediary in order to be able to provide these products for patients that need them at a much lower cost. So we're only an organization that have been around for 3 or 4 years, but we're already, I think, doing a lot of interesting things. But this slide here shows you that this class of therapies is very successful, right? And it's increasing in number. So you can see the first CAR-T approved products. mainly for the hematologic malignancies that are out there. There are some stem cell products that are now being approved for beta thalassemia, also now sickle cell disease, some genetic diseases. But we're at the letter A of a whole new field of medicine, right? And why I'm so excited about this field is we already have proof of concept of baseline that works. And this is very different than drug therapy, right? So when you try to look for a new drug, you do a screen of a library, then you have this drug, you have this drug candidate, and then that drug can fail because of pharmacodynamics or toxicity because of the drug that you use. But these therapies, we already have a baseline that works, and it's just now engineering to make them more and more efficient. And we have the whole of nature in terms of genes and elements available to us to engineer these vectors to make them very effective to cure these diseases. So it's not surprising that there is a whole plethora of these therapies in clinical development. We're focused on autologous products, right? I'm going to tell you the reason why, but let me also first tell you the way that these autologous products are produced. They're patient-specific. They're the cells of the patient that are taken out and put back in. So you can see on the top there, the cells are removed. They can either be T cells or stem cells in general. They can be other cell types as well, but mainly T cells and stem cells for many of these diseases. Then to those cells, you add a vector. That's the most critical reagent. The vector is the entity that delivers the genes into the cells, so you reprogram them. It's important to get integration into the chromosome for many of these diseases because you want the cells to expand, and when the cells expand, you want the gene to be in the daughter cells. So the gene is then delivered, the cells are reprogrammed, and then infused back into the patient. You hear a lot about allogeneic and iPS cells, and these are very interesting technologies for the future. But we believe that right here and right now, autologous will be front and center because there's still some thresholds to overcome in order to make allogeneic and iPS cells a reality. They will become a reality in the future, but it'll take a lot of work. What is working right now and what we're focused on are these autologous therapies. But as Antonio had mentioned, the price is very high, ridiculously high. Just for the product alone, at least $350,000 per patient. This doesn't include any of the clinical costs, just the product for the cells, right? And so this pricing is really not sustainable for insurance companies or health systems, even like in countries like the US, right? Right now, there's a great limitation on who gets the therapy, even in the US. If you're in an underserved population in the US, you're going to have a very hard time in getting the therapy. If you're on Medicare and Medicaid, you're going to have a very hard time to get the therapy, right? So this model is really not sustainable at this price and not feasible in low middle income countries. I think that's been highlighted, and it really restricts access at this kind of pricing. So why is the high price, right? So the high price is because companies are set up because they have to serve shareholders, right? And when they serve shareholders, they have to make it profitable, right? Because that's why the investment has been made. There's no bad players here. It's just the way the system is, right? And to do that, they need to control the product. They don't want to work too much with hospitals. They'll get the cells from the hospitals, but they want to make the product themselves. And so when you make the product yourself, you have to create a huge infrastructure for these autologous therapies. And what that means is the cells come from the hospital, an apheresis product, are shipped to a centralized manufacturing facility. Oh, sorry. Yeah, that's my fault. I sometimes talk too much. The cells are shipped to the central manufacturing facility. There they are made with a lot of infrastructure to make sure that the right cells are made and the right cells go to the right patient, back to the right hospital. That's a huge, expensive labor infrastructure. The major cost that you see on that price is the labor to make sure that that product is made correctly. And then you ship it back to the hospital. This infrastructure is very, very expensive, particularly when each company makes its own factory. Right? When each company makes its own factory, that investment has to be then come back in terms of the final price of the product. So I'm not going to go into the details. There are many reasons why. What? Oh, don't lean on the table? Oh, that's the problem. All right. All right. Good. Thank you. All right. We believe a much better solution is what we call either decentralized or point-of-care manufacturing, where you make the product locally at the hospital, right? Where the hospital themselves are making the product. It's a lot like bone marrow transplantation, right? Bone marrow transplantation is an effective way to deliver transplant products to patients. And all we're doing here, adding to that paradigm, is genetic modification, right? So what we have here is that this product can be made either within the hospital or very close to the hospital. I'm going to show you some mobile units that are made, that are placed close to the hospital, that will actually be able to make the product very close to the point of care. And this economics is much cheaper. It's 1/10 of the cost. So instead of having $350,000, you can actually make the product for about, I didn't touch it. You can make it for a 1/10 of the price, right? So that becomes economically, when you have a one-time therapy at about $30,000, that becomes an economic equation that is sustainable. And I just want to show you this slide is how the Caring Cross model is different than the pharma biotech model. So, and again, I want to emphasize there are no bad actors here, right? I meet a lot of CEOs and managers at pharmaceutical companies that would love to lower the price, but they can't because their infrastructure costs are much higher, and they have to make their shareholders a good profit because that's why the investment was made. So it's a catch-22 situation for them, right? But they focused on innovative, novel products to displace the current therapies. When you look at pharma, that's what they're really focused on. They focus on novel IP, intellectual property, right, that protects that high price and protects that market share. And they need to have that high price to maximize shareholder value because that's the reasonable reason why shareholders put the money into the company to begin with, because they want to make money of their return. And they're motivated to justify a high price and a high return, not lowering the cost. So when you see a lot of companies, when they go to meetings and they're presenting, they're talking about value. Oh, this price is high value because of X, Y, and Z. They're not thinking at all about, oh, how can we lower the price in the future? They're trying to maximize the value. Again, no bad actors, but this is the model that biotech and pharma have. And they'll only develop therapies that are fully controlled by the company and bring a high rate of return to satisfy investors. Fully controlled is important for them because if they have a model where, let's say, part of the process is done at a hospital and part of the process is done in the company, that's a problem for them. Because if you have one little adverse event, oh, that's going to affect their product. They want to have full control to make sure that that product is delivered in a way that is reproducible for them and reliable for them. And that's kind of understandable. Our model is a little bit different. We're a non-profit, and we're really focused on not novel products, but improved therapies with freedom to operate. What freedom to operate means is that you're not crossing any patent that is unenforceable patent. And the reason why we can find this freedom to operate, we've been in the therapy for 30 years. A lot of the patents are the ones that we've done. So we understand what you can do and what you really can't do. And also it's based upon our knowledgeable team. A lot of the old patents, like the CD19 patent by Novartis, is becoming expired here at the end of this year. So that leaves a lot of openness for new people, new companies, or new groups to come in and provide these therapies without any worry about infringing any patents. And there's also a lot in the public domain. A lot of this research is really done in academic centers, right? And when they're done in academic centers, they publish. Once they publish, you can't patent. And so what happens is that many companies, when they patent, they have to patent very specific sequences because the broad concept is already in the public domain. And when they have to patent specific sequences, that allows other groups to make other sequences that still are effective, but still are not infringing. So that's kind of our strategy. And then our therapies that we're trying to work with organizations to make sure that they're priced to be affordable and accessible. And we are highly motivated to reduce the cost of materials. We're constantly working on making the process shorter, right? More simplified and robust, right? Because the shorter the process, the less labor, less materials you use, and it lowers the cost, right? And the robustness makes the process easier, so you don't need to have expensive labor. You can actually have labor that is not maybe highly technical, but to be able to manufacture, test, and release the product. And our strategy is to work directly with hospitals and other organizations where the products are made locally at the point of care because the logistics there is very, very short. And we're willing to develop any therapy that brings benefit to patients. We don't really care so much about high profitability. Now, this point of care mechanism where you make things locally at the site of the hospital is becoming embraced by many regulatory authorities around the world. Traditionally, everything is centralized manufacturing. For pills and vaccines, you make them in one place and you distribute it to the hospital. But autologous therapies, because you're using the patient's cells, it's much more a better fitting model where you make it locally, especially if the process is fairly easy and straightforward. But you can see the FDA on the left there is embracing point of care and decentralized manufacturing, even for drugs. I think COVID taught us that having a single supply, right, chain is very weak, and you have to have multiple points of production in order to have a robust supply chain, even for drugs. But the sweet spot for point of care manufacturing in the middle on the top up there is really autologous patient cell production of these advanced therapies. And you just have to look at the draft guidance for industry by the FDA, showing that the FDA is very much embracing this. It's new for them as well, but they want to move it forward because they see the tremendous economic value that this kind of mechanism can bring. And it's already being done in places like Barcelona in Spain, other places in Europe, and other places around the world. So actually, the US actually may be more slow to embrace this, because in the US we have a very fragmented system, a healthcare system, but a national system like here in Brazil and other countries may be able to actually develop this point of care much more. I'm going to ask you, when you're thinking about point of care manufacturing, is can you make the product reproducibly at multiple different sites, right? Because if you can't do that, you don't have a point of care model. You have to be able to produce the product, hospital, hospital B, hospital C, the same way, right? So that's why Marcos de Lima and I, we published this paper in Nature Communications a couple of years ago in collaboration with Michael Mashan, who was a PI in Moscow, Russia, where we had two very different sites. At that time, Marcos was at Case Western Reserve University in Cleveland, Ohio. And Dr. Michael Marchand, he's a pediatric oncologist in Moscow in Russia. So we have two very, very different places, right, that are using the same device, the same vector, the same materials. And when you do all those things, when you use the same materials and same devices, well, guess what? You have a highly reproducible product. There was not that much difference between the products in Moscow, Russia, or in Cleveland, Ohio. So we proved the concept that point-of-care manufacturing really is possible. And on the right here this is one of Marcos's patients, a patient with follicular lymphoma, a 69-year-old, right? And you can see the tremendous benefit of this therapy just before and after. If you see the lighted region, that's the tumor, and then 90 days post-CAR T, you have the tumor is gone. And also the important thing here is we have an 8-day vein-to-vein time. Many of the approved products, it takes many weeks, even a couple of months, to be able to get the product to the patient. And in that particular case, many of the patients with very advanced disease die, right? Because the product is not made in time. In this case, we have 8 days vein-to-vein. So every single patient that was enrolled on our trial was treated, right? So with very good outcomes. And actually, this gives a little bit more color around the outcomes. You can see my favorite person, Marcos de Lima, on the top left up there with his team. At that time, we used the Prodigy, right? That's Michael Mashan on the right on the top there. And these are two patients on our pediatric clinical trial from Moscow, two separate families, right? And you can see the results. Sorry that the slides are a little bit off, but at Case Western with the adult B-cell lymphomas, an 82% overall response rate, right? At the pediatric center, which I can't pronounce, in Moscow, 90% overall response rate, right? And then this is a very interesting study at the Medical College of Wisconsin, and this is leading on to why we want to improve the therapy, why we don't want to just do another CD19, why we want something better. Here we had a bispecific CAR, a 1920 CAR, and we had a 92% complete remission rate and only one relapse to date, right? So that's a very interesting study because we know that about a third of all the CAR-19 patients that receive the therapy, and correct me if I'm wrong, Marcos, but they get relapse because they lose antigen. They lose the 19. When you lose the 19, well, what happens is the tumor grows. But when you have a CAR that attacks two antigens, 19 and 20, you minimize that chance of relapse. And in fact, our collaboration with Fiocruz is to develop a triple, a 19, 20, 22. So really, have very little chance of relapse due to antigen loss. And also to put on top of this is that we're really focused on this decentralized manufacturing. If a hospital has clean rooms, fantastic, just adopt the workflow. But if you don't, you can actually purchase these mobile clean room units that are clean rooms that you can put in the parking lot of any hospital, right? And then that way you can manufacture the products locally, right? So you can see here on the bottom here that we're focused on the workflow, right? And traditionally it's been 14 days. We now have a sweet spot of about 6 to 7 days, but we can see how we can make this product in 1 day in the future. This is just to show you that in Brazil, the central hub, the reference facility, will be at Fiocruz here in Rio de Janeiro. And then you have basically other hospitals that are part of the network where you're actually manufacturing and testing the product locally. So we're focused on a pipeline of products here, right? So we have an anti-leukemia lymphoma CAR. We have a CAR against HIV. They're the two first products that we're collaborating on here in Brazil. We also have a stem cell therapy for sickle cell anemia and beta thalassemia. It's very similar to the approved product. Those products are off patent, those vectors. So we now have complete freedom to operate to make exactly that same vector and make it at a low price. Make it at a low cost. Multiple myeloma, solid cancers, and I'm sorry that the slides are not rendering correctly. So the anti-HIV product is basically, we published this in Science Translational Medicine back a few years ago. It's a dual CAR. It's a CAR that binds to 2 receptors on the HIV GP120 molecule. And the way it works is that these duo CARs on the bottom there have 2 molecules, right? And the first molecule on the left binds to the GP120 molecule on the HIV, which is produced in productively infected HIV cells. So when HIV has its replication cycle, it infects a cell. Then when the cell becomes activated, one of the first things it does is expresses GP120. When it expresses GP120 on its surface, that's a unique target. That means that the CAR can see that cell and kill it before it produces a lot of viruses, okay? The second CAR on the right also acts in the same way, a co-receptor molecule it binds to, but it has a second function. It also protects CD4 cells from infection by HIV. So when HIV tries to infect a CD4 cell, which is the way HIV spreads its disease, right? It actually acts as a dominant negative fusion inhibitor. It prevents the fusion of the virus to the cell so it cannot infect it. So we have done a number of different studies. We actually had a follow-on paper in JCI Insight, but this is the bottom line here. We injected non-skid mice, first with peripheral blood mononuclear cells that are infected with HIV, and then challenge those NSG mice with human, again, CAR T cells, right? And you can see on the left there the replication of the virus in the whole body of the animal, right? And you can see in the UTD, the control animals, you're getting replication of the virus, while in the animals that contain 2 versions of the duoCAR, D13 is the one that we're in the clinic at the moment, complete inhibition of replication in the whole animal. And the other point that we showed was we showed protection of the CD4 compartment. You can see the untreated in UTD, you get CD4 depletion in the whole animal, while in the duoCAR transduced cells, you got protection of that cells. So for multi-CARs for cancer, as I mentioned to you, there's a problem with a single CAR, right? You can get relapse due to antigen loss. So we've developed these triple targeted CARs where we're targeting both 19, 20, and 22. And the way that we analyze this in an animal model is, these are all little mice. You can see them up there, and you can see that the bigger the signal, blue and red, that means that's the tumor growing in these mice, right? So the tumor grows pretty rapidly when you have a TA, which is just the control, just put the tumor alone. The tumor grows and kills the animal pretty quickly. And what we did with these mice, we injected 4 different populations of cells. The parental cell line that contains 19, 20, 22. Then for other cell lines that were either knocked out in 20, knocked out in 19, or knocked out in 22. We're mimicking that relapse, mimicking that antigen loss in these animals. So all 4 populations of cells were injected into every animal that you see there, right? So in the untransduced control, that means just normal cells put into the animal, you can see that the tumor grows quite happily. Now, if you have a monoCAR, M19, right, well, you get a delay, but you eventually get relapse, right? You see the cells grow out in the animals, right? If you have a mono 20 CAR, you can see again, eventually the cells, those 20 negative cells, grow out, right? If you have if you have a 22 monocar, you can see that eventually you get breakthrough. But when you have a triple car, you don't see that breakthrough, and we have 4 different flavors. So when this paper was published a while ago, we have now developed some new versions at Caring Cross, and we're sort of repeating some of these studies, and we hope to be able to now get into the clinic here later this year. We're completing the animal studies as we speak. And so we just have published in this area quite a bit. So a little bit of the development. So the IND submission, we're going to start in the US with a clinical trial with Dr. Marcos de Lima, right? Then once we get preliminary safety and efficacy data, then we'll initiate clinical trials here with INCA, right? So INCA will be the NCI, is the local National Cancer Institute, in which we'll be collaborating for the phase 1/2 clinical trials. Yeah, so there's 2 overarching aspects to the project, teaching Fiocruz how to manufacture the CAR T cells, and 2, how to manufacture the vector. Those are the 2 goals of the project here that we're tech transferring. And with that, Fiocruz will be fully self-sustained in order to be able to manufacture these products and collaborate with hospitals to deliver them. And these are the clinical collaborators who we start to work with. Marcos is there on the left. Martin there in the middle, and Nelson from the Einstein Hospital in Sao Paulo. These are one of the top collaborators. There are others now, but these are our top clinical collaborators. And you can see here is the protocol is now being developed. Again, noticing OSU as the US collaborator, INCA, Albert Einstein, and then also working with Anvisa. Yeah, I don't know what's going on. Yeah, this is a collaborative project that we will do co-development. We're not going to just stop with these 2. That pipeline that I spoke about earlier, this is a collaboration, and this is a chance for innovation. We actually want to collaborate with researchers here so that technology and knowledge can be resident here in Brazil, so that new entities in the future can be developed locally. And I don't understand anything what's on that page, so I'm going to give it back to Antonio.
Thank you very much, Bodo. So, as I mentioned before, a big part of this is also regulatory. And based on the Barcelona experience, and where the Spanish government developed a fast-track regulatory approval for the Barcelona group, we went to Anvisa because of a very long-standing collaboration that we developed with Anvisa through the CAT, Comitê de Assessoramento Técnico, thank you, technical collaboration, because, as you know, Anvisa had no experience in advanced therapy medicinal products or cell therapy or gene therapy. And we started a collaboration with them through the National Network for Cell Therapy. And I think this was in place since the early 20s. And through this collaboration, we gained some respect, let's say. And then we organized also a network of collaborators through what's called RENETA, Rede Nacional de Especialistas em Terapias Avançadas, and which is now headed by Adriana. And so we suggested to Anvisa that it was very important to develop a fast track for the advanced cell therapies. And because that would, if we didn't really have a way to produce those things in Brazil, that would bring SUS to bankruptcy. And so Anvisa was very sensitive to this, and in November 2022, they published this request for applications that was basically targeted to Fiocruz and Butantan. And the result of this is that you can't see, I couldn't expand it more, but the result of the RFA was that CAR T cells were approved for Butantan and Fiocruz. We have a fast track at Anvisa to bring those therapies and register those products in the shortest time possible, obviously guaranteeing the entire safety to the patients. So, as in the States, where FDA has fast track for a lot of the advanced therapy products, we now have a fast track at Anvisa also. And then from now on, I'm going to talk about the other tech transfer that's being worked. I think the term sheet is being signed this week, and it's a collaboration with the University of Pennsylvania, the group of Professor James Wilson there, who is a pioneer in vivo gene therapies. And what Jim Wilson has done is that he consolidated all the IPs that he developed, or his group developed at UPenn in this for-profit company called Gemma Bio. And the idea, there you have the number of companies that were created from the gene therapy product at UPenn. You can see there is a lot of the companies that were created. And as Bodo mentioned, if you can create a lot of those companies and you want to go into clinical trial, you have to develop your own, or you have to go to a CDMO to make your vectors under GMP conditions. And that makes the whole thing very expensive. And so the idea here is that you can see there are, the gene therapy program at UPenn supported first-in-patient studies in 15 different diseases. They raised a total of almost $500 million in support money from government and industry. And UPenn received about $318 million in licensing revenue, okay? And the idea here of Gemma Bio is to try to consolidate all of this into one company instead of having those different companies, and to try in that way to lower price. And so the idea is that you diversify and you de-risk the operation, and the focus, as I said, in the UPenn and Gemma Bio program are neurodegenerative diseases. And it's geared toward neuropathic diseases, and the lysosomal storage diseases are a main focus. Ganglioside, the neuro people help me here, but it's ganglioside M1, Krabbe disease, and the metachromatic leukodystrophy. And also a second target are the motor neuron diseases, and they do have a product they are developing for spinal muscular atrophy, which is treated by Zolgensma at 7.6 million reais, as I showed you before. And they also have products for SMA type 2 and 3, which affect not the pediatric population, but the more adult population, and the spinal bulbar muscular atrophy. So those are the 6 targets that are being considered for therapies in Brazil. And the idea is that Fiocruz, again, Biomanguinhos will produce the vectors. Now we're talking not anymore of lentivirus vectors. We're talking about adeno-associated virus. So they're going to transfer the technology for the production of the virus under GMP conditions. And those clinical trials are going to be run here in Brazil and in the rest of the world. And the technology transfer basically encompasses doing the clinical trials here, and the Brazilian government would pay for those clinical trials in Brazil, okay? And so, again, we've been to Anvisa. We talked to Anvisa about having this also on a fast track. Anvisa agreed to it under obvious safety conditions for all the patients. And although in this slide, Gemma shows that, of course, the Philadelphia hub is going to work. But other hubs would be in London, Seoul in Korea, Saudi Arabia, and Fiocruz in Brazil. But the idea is that Fiocruz would produce the vectors globally for all of those sites. And those sites require centers of excellence for treating the patients. And here in Brazil, we have a number of those centers, especially centers related to Casa dos Raros, which is an initiative led by Tony Dyer and Roberto Giuliani, who has a lot of experience in neurodegenerative diseases and has done a lot of clinical trials in mucopolysaccharidosis type 2 in Brazil. And the idea is that a number of those centers have already been contacted to set up the clinical trials here. And so that's the platform of Gemma Bio that was selected by FDA for a pilot for rare diseases, which is called the Warp Speed, okay? Jim Wilson has a very close relation to Peter Marks, who directs the cell and gene therapy program at FDA. And the idea is to, as Bora mentioned, is also to simplify clinical development and manufacturing, reducing costs and accelerating timelines. The FDA has a project manager dedicated to this. And the idea here in Brazil is that we can produce the vectors at a much lower cost than in the States or the rest of the world, is to access about 20 national clinical centers of excellence to run those clinical trials in those 6 diseases that I mentioned to you. Accelerate Anvisa approval through the fast track process, have a lot of those products approved conditionally, and the discussion about price and reimbursement is that the agreement would involve Fiocruz has total control over the price for the Ministry of Health, so we can go as low as we want in price. And for the rest of the world, we will be supplying the vector to Gemma Bio, and they set the price for the rest of the world, okay? And it's estimated that we can lower, well, as part of the agreement, it's estimated that we should reach about 20% of the US market price for the in vivo therapies. So that means lowering by a factor of 5 the price. And obviously, if, you know, the price gets lower in the States, we also lower the price here until, you know, they come to a reasonable price. I think that's the last piece of information, but I want to show you the last slide is the launch, the official launch of the program yesterday at Fiocruz. If you remember, we started working on that on 2022 and June 2022, so it's almost 2 years. We've been in contact with Boro, Marcos, Jim Wilson, discussing those things, and yesterday was the official launch with the Minister, Nízia, with the Secretary, Gadelha, Mário Moreira, the President of Fiocruz, Boro, Marco Krieger, Maurício Zuma, who is the Director of Biomanguinhos, and then Roberto, qual é o sobrenome dele, que eu nunca me lembro? Não, do INCA. Roberto Gil, the Director of INCA, and right there at the, his name I don't forget, but at the right corner is Martim Bonamino. And so it took 2 years, but I think it takes a long time. It was sometimes an excruciating work. You can imagine that to make those agreements, you have to go through all the lawyers. It's really a hard time, but I think we've succeeded. The idea is that we're getting 2 of those containers that Boro mentioned by the end of the year, probably November. And then we start the certification of the containers with Anvisa and training with the team from Boro and Marcos de Lima. And in the first trimester of 2025, we will start the CAR-T trials in Brazil using first Boro's CAR-T, and it's very important to mention here that what he's giving us and he's tech transferring is the triple CAR-T, okay? So it's different and it doesn't compete with the Butantan CAR-T, which is a CD19 mono-CAR, okay? So we're thinking about improving. There is a very good understanding between Mário and Esper Callas, who is the current president of Butantan. The idea is that the 2 big producers of biological products in Brazil are not going to compete anymore. They're going to collaborate. They're going to be complementary. And so the CAR-T at Butantan is still going on with the CD19. We're investing in a 3rd generation CAR-T, not the first. And the idea is that we think it's going to be a better product, and then we will supply Butantan and USP and whoever wants this vector, okay? So I hope I've given you an idea of what is being done. I want to thank Boro and Marcos for agreeing to come here, and especially Boro for lecturing on a way that I certainly could not do about the CAR-T for HIV and the hematological cancers. And we are open to questions. Thank you.
Professor, muito obrigado. Vamos abrir agora o espaço para perguntas.
Excelente palestra, Antônio. Eu queria fazer uma pergunta para ele, Boro. Posso fazer? Assim. very nice talk.
Yes. Hey.
I would like to know if this kind of technology, you can already treat like glioblastoma, brain, cancer brain, if the outputs that you show like 82% for the leukemia, lymphoblastic cancer, you could also have some kind of treatment in the brain tissue.
Yes, there are actually some clinical trials in that regard, and actually probably Marcos is the right person to review that. But there have been a GD2 CAR for various gliomas, and there is work going on. Crystal Makel from Stanford is doing a lot of that work. Marcos, maybe you